Hello everyone. Today we'll be taking up the last topic of biomolecules, which is lipids. Now, what are lipids? As we all know, lipids are the fats. Lipids are the fats. Now, what kind of fats are included in lipids? Lipids will include all the fats, including simple, derived, or the trans fats. So, we'll be discussing all of these one by one. Now, before discussing these, let me give you a brief introduction about what are lipids, what are their chemical and biological properties. Now, the lipids, it is a collective name which is given for a variety of organic compounds like fats, oils, waxes, or steroids that are found in the human body. These lipids are insoluble in water, but these are soluble in the inorganic solvents like alcohol, acetone, oils, ethers, etc. These are also made up of carbon, hydrogen, and oxygen. Just like carbohydrates, lipids will have the same composition, carbon, hydrogen, and oxygen. The only difference being that the ratio of oxygen is very less as compared to the ratio of carbon and hydrogen. This is the reason why the lipids give more than twice the energy as compared to carbohydrates. So the basic source of energy in our body, the stored source of energy in our body is lipids. Now, what is their role in human body? Components of these are the components of cell membrane. Cell membrane, which means that all the membranes of our cells are made up of fats. This is the most important component of our cell. Then these also act as storage cells. These act as a storehouse of energy, like the triglycerides. The triglycerides present in our body are the major source of stored fat, and triglycerides are usually stored and broken down in the liver. Then these are the chemical messengers. This is the most important role of fats in human body, that these act as chemical messengers. For example, the steroids like the cholesterol. We'll be taking up steroids in detail. Then there are some minor functions of fats like protection, waterproofing, insulation, and providing buoyancy to the human body. Now, let's see what is the major classification of lipids. Classification of lipids. Lipids can be either classified as simple lipids, these can be complex lipids, or these can be derived lipids. Now, what is the difference between these three kinds of lipids is that the simple lipids are esters of fatty acids with some alcohol, with any kind of alcohol. It can be either methanol, ethanol, propanol, or any other alcohol, but a fatty acid is mandatory. Complex lipids, these are also made up of esters of fatty acids plus alcohol, plus there would be some another group. It can be either a glycoprotein, it can be a simple protein, or any other group. So complex lipids are basically simple lipids, but with some other functional group. Now, derived lipids, what are derived lipids? These are the derivatives of either simple or complex lipids. I'll give you an example. Simple lipids are the normal fats and oils that we consume in our daily life. Then complex lipids, these are actually derived from fatty acids, alcohol, along with some other group. Example can be either phospholipid. What is a phospholipid? It is made up of normal fat plus phosphorus. It can be lipoprotein. What is a lipoprotein? It is formed of lipid or fat along with a protein group. Now, derived lipids, as we already know, that these are the derivatives of simple and complex lipids. These can be either steroids, or these can be terpenes, or these can be prostaglandins. Now, one by one, we'll discuss all these groups. But first of all, I would like to introduce a term called as esterification reaction. Esterification reaction. Now, what is a esterification reaction? As you already know that what are fats, what are lipids? Lipids are esters of fatty acids plus alcohol. So esterification reaction is basically a reaction of formation of fat. Now, what happens in an esterification reaction is that in a basic esterification reaction, we will have a molecule of glycerol, plus we'll have a molecule of fatty acid, which will give rise to the most basic type of fat, which is triacyl glycerol plus water. Now, the chemical formula for this glycerol is CH2OH, 3 molecules of this, plus fatty acid. We have RCOOH. This is also the basic fatty acid, 3 molecules of this. Here we'll get 3 molecules of triacyl glycerol. I'll show you the whole structure for this. Plus we'll get 3 molecules of water as a byproduct here. Now I'll show you how this esterification reaction works. So this is how this esterification reaction is. Here you can see RCOOH. RCOOH, this is the fatty acid. Here CH2OH is our glycerol. Now a molecule of water will be lost. How? One H will be lost from here and one OH will be lost from here. So there will be a dehydration reaction in first part, in second one also, in third one also. If the water molecule is lost from only one molecule, from only one part, then it is called as monoacyl glycerol. If it is lost from two different fatty acids and glycerol, it is called as diacyl glycerol. And if it is lost from all three of this, if it is lost from the whole structure, this is called as triacyl glycerol. So now when the water is lost, the two molecules will combine up in this way and 3 molecules of water will be lost. This is the most basic example of how a fat is formed in human body or in any other living thing. The catalyst is usually an acidic catalyst here in this case. Now, this esterification reaction will take place in all the living beings, including plants, including animals, and including all the living world except the viruses. Esterification reaction does not take place in the lower organisms of the animal kingdom. Now let's discuss some properties and types of the fatty acids. Properties of fatty acids. Properties of fatty acids. There are more than 30 types of fatty acids that are found in the living forms. Acetic acid is the most simplest kind, and the most complex type is the stearic acid. These fatty acids can either be saturated fatty acids or these can be unsaturated fatty acids. What are saturated fatty acids? Saturated fatty acids are those fatty acids in which all the internal carbon atoms possess hydrogen groups. All of the bonds are saturated by the hydrogen groups. For example, palmitic acid. Palmitic acid is the best example of a saturated fatty acid. Unsaturated fatty acids are those fatty acids in which one or more than one pair of carbon are attached to some other group other than hydrogen. It can either have a double bond or it can either have a triple bond. So because of the presence of these double or triple bond, the unsaturated fats are easily broken down at the room temperature. These have very less melting point, whereas the saturated fats have very high melting points. For example, the example of saturated fat is animal lipid, or animal fat, these exist solid at room temperature. Whereas unsaturated fatty acids, these are liquid at room temperature, and these have a very low melting point. Because these have a low melting point, these are very useful for the living beings, because living beings can easily break down the unsaturated fatty acid present in the body, and they can utilize it as energy. I'll show you how what is the structure of these saturated and unsaturated fatty acids. So here we have saturated. The saturated one, as you can see that all the carbons are carrying in the main chain, all of them are carrying a single bond, so they are completely saturated, no other group can be added here. But in unsaturated, you can see here we have a double bond in between these two carbons, which means that at this place it can be attacked and broken down and some other group can be added, because of which it is very volatile in nature, because of that it has a very low melting point. So I hope this is clear with everyone. Now coming to the next part, we have, okay. So coming to the next part, we'll have the last part, the last kind of fatty acid, which is a steroid. Steroids. Now, what are steroids? In layman's language, steroids are very complex fats which help in restoring the function of the human body. These are lipids of very high molecular weight, very high molecular weight. These are crystalline in nature. These are crystalline in nature, and these are usually made up of steroid nucleus. The steroid nucleus is a 17 carbon atom structure, and these 17 atoms of carbon are arranged in 4 rings, A, B, C, and D. So this basic structure remains the same. Basic structure of the 4 rings remains the same. The only difference lies in between, because of the free group that is present on this carbon. All of the steroids will be the same. The only difference will be because of this chain. Whatever chain is attached on this carbon will provide different characteristics to different steroids. So the most common kind of steroid that we study is cholesterol. Cholesterol is the most basic kind of steroid present in humans. It is present in our cell membranes. It is a precursor for another important steroids in humans, including male sex hormones like the testosterone, female sex hormones like progesterone, estrogen, and also in the bile acids. The bile acids are the acids released by our liver, because when bile acids will break down the cholesterol, only then the emulsification of fats and formation of vitamin D takes place in the human body. So this is the most important kind of cholesterol. So how does it look like? So this is how cholesterol looks like. This is the structure of cholesterol. As I have already told you that it is a 4-ring structure. You can see 4 rings here. These 4 rings are made up of 18 carbons in the case of cholesterol. This is ring A, B, C, and D. And here you can see there is a variable chain. So this chain is responsible for the characteristics of cholesterol. So this is all about part 1 of fats for today. In case there is any other topic that you want in detail in fats, you can always write to us. Your feedback is always most welcome. Thank you for watching the video.